So this is the third tutorial for question number 3 of your second assignment this summer. And you need to understand how to do 3 to answer 3A. In question 2 that you should have previously have done, you should have realized that we were doing something called Punnett Squares, and we were showing that 2 different alleles were being monitored to understand the question at hand. Here I have 3 sets of alleles. We're getting more complicated. So our simple Punnett square that we did for number 2 is not going to really work here. It's going to get just too complicated. I'm asking you, instead of a Punnett square, to completely answer this question based on the probability that I introduced in question 2. And that's how most of this is done genetically, probability, okay? And probability is going to use our 2 rules. It's going to use our addition rule when things are mutually exclusive. That's the common or the production of different gametes. And the multiplication rule when you have 2 independent events could happen. 2 different gametes could hit each other, as we saw. So we're not going to really use Punnett squares here to answer this question. But I want to understand that we're getting into real genetic analysis. Because as I've talked to you before, Mendelian Genetics says that every individual trait, and he was lucky that he picked pea plants and their traits mostly that were on different chromosomes, and he'd come up with his different rules that still apply today. Okay, but in order for him to come up with law of segregation and law of independent assortment, he had to have these alleles on different chromosomes. In any case, we're dealing with 3 sets of genes, okay? And we're going to assume they are on 3 different chromosomes. And this is a lot like what we do in real genetics, because unlike pea plants, a lot of traits are very complex. For instance, eye color is not as simple as a dominant and recessive allele. There's many genes that are responsible for eye color, okay? So when we look at genetic analysis, we're always looking more than just one simple allele or trait. Now, there are some disorders that you have read that are recessive in one allele or one spot on a chromosome. But for the most part, we're looking for multiple. So in this case, we're looking at pea plants again, a very simple 14 chromosome organism. And what we're going to do is look at 3 different chromosomes and look at 3 different alleles. Again, we're assuming that these genes are not linked. And what we're going to do is use our probability math skills. Yeah, I know it's weird. We're doing biology and you're dealing math. And I want you to understand something. This course that you are embarking on isn't just information, memorization, okay? It's understanding of concepts. And we're going to back up these concepts, okay, with quantitative data. And quantitative data means things that we can measure, okay, and support these concepts. The reason why we've come with our understanding of biology is through nothing more investigations and studies. As you're starting to see in your reading, there's little studies that pop up, and it's from these studies. Of course, the first investigation was what Mendel did in genetics, but we've grown from that. And from these experimental studies, from these experiments, these labs, they have measured things, and from their measurements, they have developed these concepts. So we have to deal with a quantitative aspect in biology. We have to, in order to understand concepts or vilify them or justify them, we have to measure. And you're going to see a lot of that in this course. So it may not be the course you thought it was, but that's what real life science is: investigation, measuring, seeing if it upholds what we expect and predict. So in this case, what we're going to do is we're going to take 3 different alleles. And again, we're assuming they're on 3 different chromosomes. And we're going to learn later if they're not, okay, it's not going to give what the Mendelian predictions that we get, okay? So starting this off, what they're saying is what fraction of the offspring from this cross. So what we're going to have is, let's say, a mother with this set of alleles for one trait, this set of alleles for another, and this set of alleles for another. So 3 different alleles. You can see this one's homozygous for dominant for all the 3 traits that we're studying. And we're going to cross it with, let's say, a father who is homozygous recessive for the first allele, heterozygous for the second, and then homozygous recessive for the third. Okay, now we certainly can do a Punnett square here, but it's going to get very complicated with 3. You did a Punnett square with 2. And what's going to happen is you're inevitably going to make mistakes here once it starts getting more than 2. And to be honest, Punnett squares kind of die out after 1 or 2 traits being studied because we're going to use the probability math. That's how we're going to go about doing this, and that's how you have to be a skill you must have in this course, okay, in predicting what results we should get. All right, so forget the Punnett square in terms of the large Punnett square. So what we're going to do is essentially we're going to make gametes, okay, of this one, gametes, and we're going to have gametes from the mother and the gametes from the father. And of course, they're going to come together to make offspring. Now, what combinations of these alleles will they make? Well, I'm not asking for all the combinations, and certainly that's another question for another day, but I'm asking for a particular type of combination. What fraction? Now, fraction is a probability, okay? 1/2, okay? If you think of it, if you toss a coin, you have 1/2 the ability to get a heads or a tails in the first toss. So a fraction is the probability. It's what we're talking about. Of the offspring from this cross is predicted to be homozygous recessive for at least 2 out of the 3 traits. Now it's worded kind of weird, but if you're looking for at least two, that means I'm looking for combinations of offspring, okay, that have at least 2 homozygous recessive alleles in the offspring. Notice only the father, if we make this the paternal, okay, only the father has it. But when these come together and they separate independently, okay, and using the 2 laws of segregation and a law of independent assortment, these guys will separate into gametes totally random, okay, and make offspring totally randomly, okay. And what combinations and what percentage or fraction of the offspring are going to have at least 2 recessive, okay, traits being revealed phenotypically, meaning who's going to be homozygous recessive. Now, when they say at least 2, and that's kind of an important understanding here, that means you can have 3 recessive. You can have little p, little y, little I, but you can't just have 1. You have to have at least 2 or 3. So at least 2 of these of these 3 alleles, okay, in the offspring, okay, will have be homozygous recessive, and you can have the third. Okay, so when you say, can you give me at least $10? Well, I can give you $10, I can give you $20. I can't give you 5. So at least 2 or 3 characters means I can have 2 of the 3, and I can also have all 3. And if you're not quite getting what I mean by all 3, we'll get to that. Okay, now I said we're not going to use Punnett squares, and I'm not entirely I'm not being entirely truthful here. We're going to use a little bit of Punnett squares to get us going in this. We're not going to write the big Punnett square of all 3 of these, okay, traits or genes, okay, and all the possibilities, because essentially you're going to miss a possibility when you try to do this. So when you just have, let's say, p and little p, that's easy to do a Punnett square with. But when you have a second and a third, it starts getting very, very complicated in terms of what are all the combinations, and it's easy to miss one. And that's, again, why geneticists would use the probability and stay away from these Punnett squares. And there's other reasons as we'll go on in the course. However, I'm going to just break this apart right now into small little Punnett squares so we can look at the individual alleles. Remember, this is going to occur on different chromosomes. Okay, so let's do p and little p. So let's do a simple Punnett square, and we're going to see what the probability of what offspring I can have here. So let me do p little p. Let's do the mother up top, the eggs on top, and do the sperm on the bottom. Okay, so I'm just showing you, and I guess I'll just keep it the same color because I can. Okay, so little p and little p. Okay, and this would be, let's say, the maternal on top and the sperm on the side. So the mother here, and here's the father. So what other combinations? We have big P, little p, little p, little p, big P, little p, little p, little p. Okay, a lot of p's. All right, now, what did this little Punnett square tell me? Again, I'm not doing the big, I'm just doing the individuals to get an idea of the probability. Now, understand what happened. In order for these two p's, okay, these guys will segregate, law of segregation, okay, and it'll be totally random what side of the what? Side of that equatorial plane you have in metaphase, okay, which side will be the little p and what side will be the p? Which side is going to go where? Okay, that would be the big P. So it's totally random how they break apart. And again, you need to see the big picture. So I'm just showing you the outcomes of these two alleles first as they segregate and go into gametes. Now, why did I do this? Well, I want to look at this very, very carefully. Okay, you can notice that little p and little p would occur 50% of the time or 1/2 of the time. We can say little p, heterozygous, okay, here would be 1/2 as well. So the chance of having just these outcomes is 1/2. Now you say, well, okay, I'll get that. Now understand this. This 1/2 equals 1. These are mutually exclusive each other, right? There's only 4 outcomes. There's either going to be a big P, little p, or a little p, little p. Those are the only 2 combinations that can be made from the segregation of these 2 alleles into a new organism. Okay, that's the only 2 combinations. I hope you can see that. Okay, and we're going to use this 50% or half probability later on. Notice they add up to equal 1. They are mutually exclusive. They exclude all other independent events, but they're based on each other. They equal 1. Okay, let's do the next, okay, set of alleles from the mother and the father. Let's put the mother up top. We've got Y, little y, and the father, Y, little y. Why? Because I said so. Why? Because I'm not really funny right now. Okay, so let's break. So what we get is they're both heterozygous. So big Y, big Y, big Y, little y, big Y, little y, and then little y, little y. Sometimes I confuse myself. Okay, so what do I have here? Well, I have a 1/4 chance when I put these 2 alleles together, when they segregate, law of segregation, these 2 chromosomes, the genes in the chromosomes separate. I have a 1/4 chance of this happening. I have a what? A 1/2 chance of that happening. And of course I have a 1/4. Notice they add up to 1 because these are mutually exclusive. If I was to add these up, they equal to 1. The production of organisms from gametes, knowing exactly what the mother and father have, this is going to be an addition rule. This is how we would add up. They should equal 1. These are the only combinations, just like a what? A coin. You can either have heads or tails. 1/2 will be heads, 1/2 will be tails. You add them together, that equals 1. 1 equals all the possibilities. These are all the possibilities. Okay, let's pick another color because I'm into colors today. Magenta. Let's do the last trait. Okay, and don't know why that was colored there, but there it is. Let's make that magenta. And what we have is from the mother, we have big I, little I, and then we have little I and little I. Okay, let's do our simple Punnett square. And again, I'm only doing the one for each other to know what the possibilities are for each of them. And if you were to do this many times over, you could probably look at these and know, but right now, we're going to do this. So big I, Dominic goes first. I, little I. Then we have little I, and little I, little I. And you can see that the heterozygous combination is 1/2, and the what? The homozygous recessive is 1/2. Of course, they equal 1 because these are all the possibilities. They're mutually exclusive of each other. Okay, so now, knowing that, I'm going to start putting together the entire, okay, combinations. Now, let's just kind of rewrite. We had P, little p, big P. I'm just going to rewrite what I had here so we can see what we're doing. We had P and little p, and I think it was homozygous recessive. My short-term memory is going at this height. Okay, little p, and I'm just going to just rewrite that so I see it. Okay, big Y, little y, and what was the other one? Same thing, I should remember that. I guess I can read it from the boxes ahead, and just afraid to make a mistake here. I, big I, and then a little I, okay. So this is our cross. Okay, so what are our possibilities? Now we're not looking for all the possibilities. Okay, all I'm looking for is what fraction of the offspring are predicted to be homozygous recessive. That means 2 of these for at least 2 of the 3 characters, which means we can have 3 recessives. Okay, so let me talk about what they want here. So when these cross, and here are our possible combinations, I'm looking for 2 out of the 3. So let's start with one possibility. When PP of the mother, okay, segregates and makes gametes to make PP of the father, one combination is big P, little p. So let's write that. Big P, little p. Boom, there it is. Okay, now, if this is going to be heterozygous, and I said to you in the problem, I want what? 2 out of the 3 to be homozygous recessive, then the only other combinations that would work that I'm looking for would be little y, which is a possibility, and II. You notice these have 2 recessive, at least 2 homozygous recessive. That's what we're looking for. We're looking for combinations that have 2 homozygous recessive. Okay, now we have PP. Another possibility, so I'm going to write it. So PP, that's another possibility when these come together. And now we're going to have PP. And now if this is at least, this is homozygous recessive, so I could go YY, there's my 2 out of the 3, and make this I. So you really have to think here. I'm looking for 2 out of the 3 possibilities, and these are my possibilities. They're in, notice these possibilities here, and here, and here are independent of each other. In these little Punnett squares, I was dealing with the law of segregation, okay? Now when I compare these to each other here, this is the law of independent assortment. These things happened independently of each other if they happened on different chromosomes. That's what I'm doing here. But we got to put these together. So now, 2 out of 3. Well now, wait a minute, didn't I also say that I could have 3? So if I'm going to use PP, finish the string, YY, I can also have II as a choice. Now, independently, I could have little p. Independently, I can have Y, and independently, I can have II and II. And that kind of runs a string. Okay, let's go to the next one. Now, if we want to find all the combinations, there's probably some other ones that I could do. All right, let's think here. I've done the PP All right. Could I do homozygous for the P and the P? I'm looking for 2 out of 3. All right. Could I do big Y? Big Y? And then II? All right, 2 P's independently. This becomes Y and Y independently. This becomes little I independently. That's Mendel's law of independent assortment. The things that you're reading, people, are coming to life. You have to not just read and take notes. You've got to internalize this. This is what I'm asking you to do. Okay, it's getting exciting. Maybe not. Okay, now, what other combinations do I have? Well, I need to have 2 out of 3 or 3 out of 3. I already have the 3 out of 3, so everything left is 2 out of 3. Okay, so I had PP, big Y, II. Why can't I have P and P and the heterozygous Y, little y, II? I don't think I have that one. So PP, big Y, little y, and yeah, you're going to have to think. You really do. It's not about reading and just, definitions. Your 8th grade science classes, okay, you've taken, I don't care if it was the Regents in Earth Science, is nothing like this. You have to think. Now, I don't think there's any more combinations that I can do here. And you can work them out. You can think them through. You can do trial and error. There's no exact way to do this. There's no exact way to think. Okay, I'm giving you the way that I break it down, but maybe you see this differently. And I'd love to hear it, how you would do it differently. But now we have our 5 possible combinations. I can't do it any other way. If I were to try to do Y, then I have the 2 sides, and yeah, I've got it all done. Okay, there's all 5 possible combinations. Okay. All right, so here's what I'm going to do next. I'm going to rewrite these. Okay, so all I did was take our 5 possible outcomes that the cross between this mother and the father that would give me at least 2 of the traits of the 3 that we're studying to be homozygous recessive. Are there more outcomes? Yes. I don't care about those outcomes. That's why we're not doing a Punnett Square. We're just going to do. Now, of course, 2 reasons. I don't care about the other outcomes. 2, it's easy to make a mistake to do a Punnett Square with 3 variables. Okay? If you can bang it out and do that, fine, but you have to be able to use these probability rules, okay, and mathematically. So here's what I'm going to do here. What we're going to take is the 5 possible outcomes, okay, and here they are. Just wrote them in a column now. We're going to use our math here to figure out what is the fraction that give us this from this cross of, of these 2 seeds. What are the percentages or fraction of the offspring that will at least have 2 out of 3 that are homozygous recessive in those traits. Okay, so how do I do that? Well, now I'm going to go back to understanding that to find the total probability, I would take the probability of P and P coming together. Well, for P to P to come together, which is called law of segregation, okay, randomly from different mother and father, and going into gametes, okay, that random separation and reforming, okay, if you notice, P and P is 1/2. So PP would be 1/2. There's a 1/2 chance. If you notice right there, we did that little Punnett Square to see that. There's a 1/2 chance of making a heterozygous, okay, offspring from that mother and father. Right there. Now, I'm going to now look at the chance, which is totally independent of P and P. That's the independent assortment law that Mendel came up with. And what are the chance of Y and Y coming together? Well, notice this is 1/4. Okay, so this is 1/4. And you can see that 1 out of 4 possibilities with these 2 sets of alleles, okay? And then what is independently, how did I and I, what is the probability of I and I being made from these sets of alleles from mother, and father. And of course, you can see that it's 1/2, as we've already looked at. So 1/2 is the probability of I and I coming together. Now, do I add or multiply these? The question is, are these independent events? Hello, law of independent what? Assortment. Okay, yes, you're going to multiply these. This coming together has nothing to do. Flipping the first coin heads has nothing to do with flipping an entirely different coin heads. You'd multiply these together, okay? So this is multiplication. So 1/2 times 1/4, well, 4 times 2 is 8. 8 times 2 is 16. Multiply the top numbers together. You would have a 1/16 of a chance of getting this exact outcome, okay? Totally, all right? Now let's do that for each one, and that's what you're doing here. You're using the law of probabilities, that mathematics we talked about, to do this. So without further frost, let's continue. To get PP, little p, what's the probability? 1/2. Independently, Y to Y to come together from a different chromosome, 2 different chromosomes. What are the chances of these independently assorting, okay, in metaphase, in meiosis, and then coming together, okay, as different gametes? Okay, what's the chance? Well, YY, okay, is 1/4, as we just saw here. And we're going to start going a little faster as we do this. And then what's the chance of big I, little I coming together independently? That's 1/2. Okay, and we do the same thing. Because they're the same numbers, it equals 1/16. So the possibility of this 3 genes, or this genotype, I should say, this arrangement of chromosomes with these alleles, okay, from this cross of mother and father is a 1/16 chance. And that's what you're doing for all of them. Okay, so little p, we already know it's 1/2. Little y is 1/4 of a chance. Little I is 1/2 of a chance. And I'm just going to multiply them because they're independent. And you should understand why they're independent. They're on different chromosomes. The chromosomes line up. If you can visualize meiosis from your sketch, different alleles on different chromosomes, okay, are going to independently. There's a reason for my madness. Okay, so you should start to see this, not just as numbers, as a problem, but see how this problem is mixing in with your other things that I've done here. Okay, so 4 times 8, it's another 1/16. Okay, and we're going to go a little faster because it should be getting easy. Little p is a 1/2 chance times the independent y, big Y, which is 1/4. Just making sure I got that right. Big Y is... No, big Y is 1/4. The Y's always mess me up. My lowercase y's have to work on. And little I is 1/2. All right. And you get a 16th again. And then we're going to do little p, 1/2. And then we're going to do big Y, little y. Oh, heterozygous here. Make sure we look. Heterozygous is 1/2. All right, 1/2 the possibility. So that would go 1/2 here. Little I is 1/2. Do the math here. Do the fandango. 2 times 2 is 4. 4 times 2 is 8. 1/8. Okay, which is the same thing as what? 2/16. All right, and what you're going to do now, because what was the question about? What was the question? If you have to go back to the question. What fraction of offspring are predicted to be homozygous for recessive for at least 2 of the 3 characters? 2 of the 3 characters. okay, which includes 3. Well, these are all our 3 possibilities, so we want to know the total. Now, remember, don't get confused. We multiplied these from different alleles and they came together, but these are all of the what? These are all the possible outcomes, excluding the other ones that we didn't have there that we would come up with a Punnett square. We're only after these possible outcomes, but it's either going to be what? This one, or this one, or this one, or this one, or this one. There's only 5 possible genotypes that we can get if we want at least 2 recessive homozygous alleles. So these are mutually exclusive to each other, so we're going to add these together. Okay, so you add them together, and you get 1, 2, 3, 4, 5, 6. You get 6/16. Okay, and if you reduce that, or 3/8, neither one is correct. So you have a 3/8 chance between the cross of this mother and this father to come up with at least 2 of the what? Traits, including 3, that'll be homozygous recessive. And that, my friends, is your answer. It's not just a problem. You have to internalize this problem and think with me how I came up with it using all of the tools that this unit, these 2 units gave you. It's not just reading, okay? And it's not just going to be vocabulary words and regurgitating. It's understanding the concepts and being able to predict and analyze and count, okay? And hypothesize using mathematics to support our hypotheses. Hope this helped.